Students, have you ever thought about where and how the structures like roots, stems, leaves, flowers, fruits and seeds arise, and that too in such an orderly sequence? You must have also seen that trees continue to increase in height and girth over a period of time. This is all due to the fact that all plant organs are made up of a variety of tissues or cells, and all these cells of a plant are descendants of older cells. The question is, then, why and how do they have different structural and functional attributes? Basically, development is the sum of two processes, growth and differentiation. So, it's essential and sufficient to know that the development of a mature plant from a zygote or fertilized egg follow a precise and highly ordered succession of events. During this process, a complex body organization is formed that produces roots, leaves, branches, flowers, fruits, and seeds.
Dear students, our today's topic will revolve around growth and development happening in a plant life. ਇਸ ਦੇ ਨਾਲ ਅੱਜ ਅਸੀਂ seed germination, photoperiodism ਆਦਿ ਬਾਰੇ ਵੀ ਵਿਚਾਰ ਕਰਾਂਗੇ। ਪਰ ਇਹ ਸਭ ਜਾਣਨ ਤੋਂ ਪਹਿਲਾਂ ਆਓ ਟੌਪਿਕ ਦੇ ਲਰਨਿੰਗ ਆਬਜੈਕਟਿਵਸ ਜਾਣ ਲਈਏ।
Upon completion of this topic, learners will be able to define growth, list characteristics of plant growth, discuss the phases of plant growth, define two types of seed germination, define photoperiodism, explain different types of plants according to photoperiodic requirements.
Hello students, ਅੱਜ ਦੇ ਪਾਠ ਵਿੱਚ ਤੁਹਾਡਾ ਸਵਾਗਤ ਹੈ। Growth is regarded as one of the most fundamental and conspicuous characteristics of a living being. It is the result of wonderful coordination of biophysical and biochemical processes at different levels in an organism. The net result is an irreversible increase in mass, weight, or volume. So let us discuss in detail what is growth. Growth can be defined as an irreversible permanent increase in size of an organ or its parts or even of an individual cell. Generally, growth is accompanied by metabolic processes which require a lot of energy, as plants, like other organisms, are made up of cells. Growth involving an increase in cell number by cell division and increase in cell size takes place. Plants have indeterminate growth and as long as they live, continue to add new organs and tissues. Growth may involve one or more of the following: increase in the amount of protoplasm, increase in the size of cells, their number, organs, and organism as a whole, increase in number of cell organelles, increase in weight. Plant growth is basically localized, a complicated process and consists of different phases. During growth, meristematic cells pass through the following three phases.
Phases of plant growth. These three phases are cell formation phase or formative phase, cell enlargement phase or elongation phase, cell differentiation phase or Maturation phase. During cell formation phase, the meristematic cells divide to form new cells. Division is restricted to the meristematic region, like apical meristems, both at root and shoot tips. The newly formed daughter cells are thin-walled and have dense protoplasm and a large nucleus. The daughter cells have the same number of chromosomes and the same genetic constitution. During cell enlargement phase, the newly formed cells absorb water by osmosis, resulting in the increased turgidity and expansion and dilation of the elastic cell wall. Cell differentiation phase or maturation phase occurs a little lower down the zone of elongation. Here, the cells start maturing to obtain their permanent size. The elongated cells develop into special or particular type of cells by undergoing structural and physiological differentiations.
So this was all about phases of plant growth. Now let us learn about seed germination. Seed germination. Students, I hope you remember which part of flower turns into seeds. Yes, ovules present in the ovary mature and become seeds. The seeds are carried away from mother plants by various agencies. After separating from parent plant, it is time for the seeds to sprout or germinate. You have seen that common garden seeds germinate if given just water and reasonably warm temperature. Basically, germination is the onset growth of a seed, often following a period of dormancy in response to suitable environmental conditions. So we can here define seed germination as emergence of radicle by rupturing seed coat. The root is normally the first structure emerging from the seed, growing rapidly and absorbing the water and minerals from the soil. Much of the water is transported to the cells in embryo. As its cell elongate, the stem pushes out of the soil. There are two types of seed germination. Let us check it out.
Types of seed germination. There are two types of seed germination: epigeal germination, hypogeal germination. Let us first discuss epigeal germination. In this type of germination, the cotyledons come above the soil surface into air and light due to rapid growth and elongation of hypocotyl. The cotyledons are green and make food to be used by stem during growing season. They finally dry off and seedlings become independent, as shown here in the visual. Example of plant showing this type of germination are cucurbita and beans. Hypogeal germination. In this type, the cotyledons remain in soil or just near the surface. Here, the epicotyle elongates, pushing plumule upwards. Cotyledons do not turn green and gradually dry up and fall off, as shown here in the visual. Example of plants showing this type of germination are gram and maize.
So these were the two types of seed germination. The basic difference between them is that in hypogeal germination, the cotyledon, also called as seed leaves, remain below the surface of the soil and the shoot grows outward, whereas in epigeal germination, the cotyledons come above the ground along with growing shoot. ਇਸ ਤੋਂ ਇਲਾਵਾ ਮੈਂਗਰੋਵ ਪਲਾਂਟਸ ਵਿੱਚ ਸਪੈਸ਼ਲ ਤਰੀਕੇ ਦਾ ਜਰਮੀਨੇਸ਼ਨ ਹੁੰਦਾ ਹੈ। ਮੈਂਗਰੋਵ ਪਲਾਂਟਸ ਮਾਰਸ਼ੀ ਏਰੀਆਜ਼ ਅਤੇ ਸੀ ਕੋਸਟ ਤੇ ਪਾਏ ਜਾਂਦੇ ਹਨ। ਇਨ੍ਹਾਂ ਵਿੱਚ ਹੋਣ ਵਾਲੀ ਜਰਮੀਨੇਸ਼ਨ ਨੂੰ ਵਿਵੀਪੇਰਸ ਜਰਮੀਨੇਸ਼ਨ ਦੇ ਨਾਂ ਨਾਲ ਜਾਣਿਆ ਜਾਂਦਾ ਹੈ। Let us see some of its visuals.
Viviparous germination. In viviparous germination, the seeds germinate while still attached to the parent plant. The embryo grows out of the seed and then out of the fruit and projects from it in the form of a green seedling, displaying the root and the hypocotyl. Due to the increase in weight, the seedling separates from the parent tree and falls into the water and develops lateral roots. Vivipary is seen in Rhizophora, Cercops, Candelia and Soneraceae.
Students, in some seeds, such as in tobacco plant, light is also necessary to trigger the germination of seeds. So plants vary greatly in response to light with respect to seed germination. Seeds which respond to light for their germination are named as photoblastic seeds. Non-photoblastic seeds germinate irrespective of the presence or absence of light. For the light-sensitive seeds, the red region of the visible spectrum is most effective for germination. The far red region reverses the effect of red light and makes the seed dormant. The red light and far red sensitivity of the seeds is due to the presence of a photoreceptor pigment called phytochrome. After studying the seed's germination, let us now focus on photoperiodism. In most latitudes, there are seasonal changes in the length of the photoperiod, that is, changes in the length of the day. There is an advantage for organisms to be able to anticipate seasonal changes so that environmental conditions are most suitable. For example, producing flowers at the right time of year to attract pollinators or migrate to avoid the harsher conditions of winter. So most seasonal events are triggered by a photoperiod of a certain length. This specific photoperiod length is basically the photoperiodism. In 1920, two employees of the US Department of Agriculture, W. Garner and H. A. Allard, working on Maryland mammoth, a variety of tobacco plants, established the fact that it was the length of the day which controlled flowering in these plants. Garner and Allard suggested the term photoperiodism to designate the response of organisms to the relative length of the day and night, and photoperiod to designate the favorable length of day for each plant. So let us see what actually is photoperiodism.
Photoperiodism. It can be defined as the physiological response of an organism to the periodicity and duration of light and darkness, which affects many processes including growth, flowering and germination, is known as photoperiodism. So, in simple words, it means that flowering in certain plants depends not only on a combination of light and dark exposures, but also their relative durations. This response of plants to periods of day-night is known as photoperiodism. The sites of perception of light-dark duration are the leaves. Photoperiodic flowering plants are classified into three types. These are short-day plants, long-day plants, day-neutral plants. Let us discuss these types one by one. Short-day plants, SDP. Flower when the day length is less than a certain critical length, say 12 hours. If these plants are exposed to day lengths in excess of this critical point, they continue growing vegetatively. Or simply, we can say that in these plants, flowering is favored by short days and comparatively longer nights. Common examples of these types of plants are Nicotiana tabacum, strawberry, Oryza sativa, Chenopodium, cocklebur, and chrysanthemum. Long day plants, LDP. Long day plants are those plants which require a long period of exposure to light in order to produce flowers. Usually, 16 hours of light are given in laboratory to such plants as compared to 8 hours of darkness. Common examples of these types of plants are Pisum sativum, Triticum aestivum, Beta vulgaris, Allium cepa means onion, Spinacia oleracea means spinach. Day-neutral plants. These plants flower in all possible photoperiods, ranging from a few hours to 24 hours of uninterrupted light exposure. We may say that flowering in such plants is not based on day length. They are also called photoneutrals or intermediate plants. Common examples of these types of plants are tomatoes, cucumbers, sunflower, maize, and cotton.
The terms long day or short day plants are actually misnomers. Research has proved that when the long night period was interrupted by a brief exposure to light, the plants failed to flower. So the scientists concluded that what is essential for flowering is long and uninterrupted dark period rather than a short day length. A brief interruption of the dark period with light nullified the effect of long night. Let us understand this with the help of a visual. Let us first examine the results with the help of a plant poinsettia, which is a short day plant. Here you can see that short day plants flower when the day is shorter than the critical length and when the night is longer than the critical length. When the night of correct duration is interrupted by a brief flash of light, the plant fails to flower. So interruption in the night blocks flowering. Now we turn our attention to a long day plant for the same test. You can see here the long day plant flowers when the day exceeds the critical light and fails when it does not. When you interrupt the night with a flash of light, indeed this plant can flower with short days if you cut the long night into short nights. So the long day plants are in fact the short night plants. Students, you must have observed here that the night interruption works fine, but the day interruption does not change the responses. So short day plants were called long night plants and long day plants were called short night plants. So students, ਅੱਜ ਲਈ ਇੰਨਾ ਹੀ। ਹੁਣ ਸਮਾਂ ਹੈ ਜਲਦੀ ਨਾਲ ਪੂਰੇ ਟੌਪਿਕ ਨੂੰ ਸਮਰਾਈਜ਼ ਕਰਨ ਦਾ। Growth is regarded as one of the most fundamental and conspicuous characteristics of a living being. Growth can be defined as an irreversible permanent increase in size of an organ or its parts or even of an individual cell. There are three phases of plant growth: formative phase, elongation phase, and maturation phase. Emergence of radical by rupturing seed coat is called seed germination. There are two types of seed germination: epigeal germination and hypogeal germination. The basic difference between these two types are that in hypogeal germination, the cotyledons, also called as seed leaves, remain below the surface of the soil and the shoot grows outward. Whereas in epigeal germination, the cotyledons are brought outward above the ground along with the shoot. Then we discussed about viviparous germination in plants. Vivipary is seen in Rhizophora, Cercops, Candelia and Soneraceae, etc. Plants vary greatly in response to light with respect to seed germination. Seeds which respond to light for their germination are named as photoblastic seeds. Non-photoblastic seeds germinate irrespective of the presence or absence of light. Thereafter, we discussed about the term photoperiodism to learn the response of organisms to the relative length of the day and night. On the basis of physiological response of plants to the periodicity and duration of light, they are of three types: short day plants, long day plants, and day neutral plants. Examples of short day plants are chrysanthemums, strawberry, Nicotiana tabacum, Chenopodium, etc. Whereas examples of long day plants are Pisum sativum, Beta vulgaris, Allium cepa means onion, Spinacia oleracea means spinach, etc. There are some plants which we discussed in the category of day neutral plants do not follow fixed photoperiods. They may bloom in any length of photoperiods ranging from a few hours to 24 hours. They are also called photoneutrals or intermediate plants. Examples of such plants are tomatoes, cucumbers, sunflower, maize and cotton. So students, enough for today. Let us quickly check with some short questions what you have grabbed today. Be attentive, here are some questions. And the first question is, what is growth? The answer is, growth can be defined as an irreversible permanent increase in size of an organ or its parts or even of an individual cell. The next question is, name the three phases of plant growth. Answer: the three phases are cell formation phase or formative phase, cell enlargement phase or phase of elongation, cell differentiation phase or phase of maturation. Next question. What is cell formation phase or formative phase? The answer is, during cell formation phase, the meristematic cells divide to form new cells. Division is restricted to the apical meristems both at root and shoot tips. The newly formed daughter cells are thin-walled and have dense protoplasm. My next question is, what is the function of cotyledons? Answer. The cotyledons store food for the baby plant inside the seed. Next question. Define seed germination. The answer is, emergence of radicle by rupturing seed coat is called seed germination. Next question is, give 2 examples of Hypogeal germination. The answer is, cucurbita seeds and bean seeds are the examples of Hypogeal germination. My next question is, what is hypogeal germination? Answer. Hypogeal germination is the type of seed germination where the cotyledons remain under the surface of the soil and or inside the seed itself. Next question. Give examples of viviparous germination. Answer. Vivipary is seen in Rhizophora, Cercops, Candelia and Soneraceae. The next question is, who suggested the term photoperiodism? Answer. In 1920, 2 employees of the US Department of Agriculture, W. Garner and H. A. Ellard, suggested the term photoperiodism. Next question. Define photoperiodism. Answer. The physiological response of an plant to the periodicity and duration of light and darkness, which affects many processes including growth, flowering and germination, is known as photoperiodism. The next question is, what are short day plants? Give 2 examples. Answer. Short day plants or SDP flower when the day length is less than a certain critical length, say 12 hours. Common examples of these type of plants are Nicotiana tabacum and Chrysanthemum. My next question is, what are long day plants? Give 2 examples of such plants. The answer is, long day plants flower when the day length is greater than a certain critical length, say 12 hours. The examples are Pisum sativum and Allium cepa. The next question is, what are day neutral plants? Also give examples. Answer. These plants flower in all possible photoperiods ranging from a few hours to 24 hours of uninterrupted light exposure. They are also called photoneutrals. Common examples of day neutral plants are tomatoes, cucumbers, sunflower, maize and cotton. Hope I have made a successful attempt to clear your concepts regarding plant growth, seed germination and photoperiodism, which will help you in fetching good marks in the exam. Thanks for your cooperation and attention. Looking forward to the next class. See you then. Thanks.